For everyone with an interest in MASH, or more broadly, steatotic liver disease, Surf's Up, Season 5, Episode 24 of Surfing the MASH Tsunami, our look from 3 different perspectives back at the EASL Congress and what it means for people's insights, strategies, and how they do their business going forward, starts now.
Today on Surfing the MASH Tsunami.
The community has done in the past to convince FDA that NASH serine stages were the right endpoint for interim approval. We will have to do the same for non-invasive tests. The case would probably have to be very strong for F2, F3 moderate fibrosis.
We can take these huge data sets and begin to not just analyze and not just be kind of like the philosophy student looking for meaning, but actually take something from it and use it for a deliverable and outcome that helps improve patients' lives in some way.
Eradicating fibrosis always had 2 pieces to it. It seems to me that number 1 is, can you put your foot on the hose, thereby stopping or slowing the rate at which patients flow into fibrosis, and then can you get better drugs to treat them once they get there? You can see that both pieces are in place.
What I come back to is the importance of tailored care. Physicians really should look at each patient as an individual. Patients are individuals and need individual care.
Ask your patient, are there some issues you want to discuss? I have some, but do you have? And then you can move on with issues that are for patients very important and listen more to patients.
There's an old organizational management saying that people do what's inspected, not what's expected. So if what's inspected is that I'm doing everything I've got to do on my checklist, and that takes all 15 minutes, and no, I don't have the time to think about the patient, it would be helpful to make sure that part of that checklist is José's question.
Last month, over 7,000 hepatology stakeholders convened in Milano for the EASL Congress 2024. This week, join pricing and forecasting guru Roger Green as he interviews PharmaNest founder and CEO Mathieu Petitjean and Global Liver Institute Vice President, Liver Programs Jeff McIntyre, about what each considered key takeaways from Milano, plus a special review of answers to last week's question of the week, all this week on Surfing the MASH Tsunami.
Remember to keep listening after the episode for this week's question of the week, sponsored by Madrigal Pharmaceuticals.
Hi, this is Roger. This week's episode introduces a new element for future episodes, plus 2 one-on-one interviews. The 2 interviews with frequent guest Jeff McIntyre of the Global Liver Institute and first-timer Mathieu Petitjean of PharmaNest address 2 questions: what they consider the major takeaways from the Milan EASL Congress from their perspectives, and how this is affecting their work and their future planning. All this is part of our effort to prod listeners into not only learning the interesting findings from major meetings, but also to explore what a former coworker used to describe as, and I quote, the meaning behind the meaning. The third feature is something we plan to make a regular part of the podcast, a discussion of the previous week's question of the week, assuming that we get answers from the audience. Last week, our question of the week stemmed from the patient advocacy session at EASL. We asked what people thought would make patient-physician communication better, and we received 3 answers, 2 from patient advocates and 1 from a public health professional in South America. Patient advocates Mike Betel, a co-chair of the EASL event panel, and José Willemse, a panelist at that event, joined me to review the 3 answers and share impressions about what they mean in the context of the broader issue. I'll discuss how we plan to share question of the week discussions for future episodes when I share the question of the week for this coming week. One thing to know up front, if your question is part of the discussion, we will invite you to join us for what will probably be your debut on Surfing the MASH Tsunami. And in any case, unless you ask us not to, we will use your name on the broadcast and give you recognition. At any rate, enough for that later. Right now, our episode starts with the conversation with Mathieu Petitjean, founder and CEO of PharmaNest. So our first interview in the series is with Mathieu Petitjean, the founder and CEO of PharmaNest. Matt, how are you this afternoon?
Hi, Roger. I'm very good. Thanks for having me here.
This is audio, so you can't see, but Matt is wearing the most interesting pair of, I guess, red, would you call them, reading glasses I've ever seen?
Yes, red in the front and blue and red on the side with the pink flowers.
Okay, and he just showed me one that was yellow in front.
Oh, they're just plain yellow.
Oh, they're just plain yellow, okay. So he's wearing the more creative, interesting reading glasses as we get together. I've had the pleasure of spending time at several meetings over the last few years with Matt, including one where we walked all the way from the meeting center to the wrong hotel to walk all the way back to the meeting center, which is where the right hotel was, which is, I think, where we really got to know each other. Matt, since you've not been with the podcast before, do me a favor, take a minute or two, just tell the audience a little bit about what you do right now and your career and academic history, how you came to be doing this. Your floor, my friend.
Yes, well, thank you so much. So first, as you can see from my French accent, I'm from Tennessee. I moved from Paris to Nashville 25 years ago with GE Healthcare. I'm an engineer specialized in medical imaging. And since then, I've done many things, including leading companies in digital pathology. I worked with the team to take the FibroScans through the FDA. And most recently, I was aware of the challenges to quantify fibrosis in liver biopsies. So we came up with the team on an image analysis method that actually seems to work to quantify fibrosis and associated histological features. We are based in Princeton, here in New Jersey, and the computer science team and AI team is based in California. So we've been around 4 years. You asked me to share something that people don't know about me. I wouldn't guess about me. So I thought we would meet in New Hope. You live not too far away from me, and I was ready to ride my Harley Davidson to go and meet with you. So for those who will meet me one day face to face, we can either talk about French wine or digital pathology or Harley Davidsons.
I have family members who ride Harleys. And for those who don't know, we've talked a little bit from time to time about New Hope. New Hope is, among other things, a really big biker town. A couple hundred bikers, usually on a pleasant weather, will show up here on Saturday. They've got a few places they go to that are their favorite haunts. And in fact, a London-based restaurant chain of biker-themed restaurants, they've got them only 4 or 5 around the world, called ACES, just opened in New Hope as their first U.S. location because they figured it had a number of affluent bikers, or at least moderately affluent bikers, who come to town and could be the linchpin for their restaurant. I don't know how it's going. I've not been there yet. I'm a cyclist, but I'm not a biker. So with that, as I said in the introduction, we're really asking for 2 questions today, which is looking back on the EASL Congress from a couple of months later, what each of our people were spoken with, what you took out of the meeting, that was A, relevant to MASLD, and B, relevant to your own work. And then we're going to talk about how the meeting has changed either what you're doing now or what you're planning to be doing so that people can get, we've talked for the last month about what were some of the lessons. Now let's talk about how they play out in people's careers. So let's start on question 1, Matt. Takeaways from EASL.
Yes, well, just before I share my takeaways from EASL, I'm going to explain what we do so it's simple as a way of setting the stage. So PharmaNest is a company that is specialized in digital pathology. And 4 years ago, we put down the hypothesis that the histological phenotype of fibrosis should be quantified in a high resolution, sophisticated way. And we also hypothesized that fibrosis looks different according to severity, which we all know, but according to other phenotypes, clinical phenotypes could be the gender, could be the etiologies that yields to fibrosis. So, the big idea here is that fibrosis equals phenotype. So we developed this tool, which requires highly sophisticated computational tools. We are a single fiber analysis company, and by single fiber, try to understand and anticipate the analogies with technologies such as single cell analysis. So we are both spatially resolved technologies, and all the data we generate on single fibers, we use them to find things that change as the phenotype change. So this hypothesis was original 4 or 5 years ago. It was not the common way of looking at scoring for fibrosis, and it happens it works very well, not only in liver, with other indications. So we do have now biomarkers for fibrosis. They are continuous biomarkers. They are under review by FDA and being used in phase 2 clinical studies. And what they do mostly is being able to identify changes within categorical stages. So for instance, if you have a patient that is in an F3 plus stage that moves down to F3 minus, when you use categorical stages, that patient would not be a responder. It is going from F3 to F3 non-responder. And we all know that through biomarkers or other markers, we know the patient has improved. We resolve that challenge. We can measure changes inside categorical buckets. We are still based on biopsies. And with that said, I'll share a little bit what we took away from EASL. For me, and then for my team, we debriefed internally. This was perhaps the most significant EASL that we've been through since even before COVID. COVID, and it's driven by several things. Obviously, the fantastic news that were reported at the late breakers. We had several studies with amazing antifibrotic and antisteatotic effects. So I think this was highly energizing for the field. For us, this was the big news, and we were excited to see that happen and to see, folks from fantastic biotech companies sitting side by side with us, and they were very, very happy to present those results. What we saw as well is the commercial deployment of Madrigal and Resmetirom. I think they're doing a fantastic job. I was very impressed by the training sessions that hepatologists have been giving to really explain how to evaluate patients and how to put them or not to put them under Resmetirom. I think this was extremely well done, and I don't think we've seen anything like that in prior EASL meetings. So these two things stand out. Now, all that said, we see as well, and this is particularly important for us, more and more convincing and significant results on non-invasive biomarkers, which we all know, biopsies as part of the criteria to enroll patients. And, if I'm a patient and somebody says, Mathieu, we're going to give you the choice to go into an investigational product or Resmetirom, right? If you want to go into this investigational study, you have to take a biopsy. If you take Resmetirom, you don't. That poses a true problem. Okay. Is that patient going to consent to an investigational study? I don't have the answer, but for me, it's a big question mark that I think needs to be addressed. We can talk about that. And the other thing that we've seen is that the effects, the magnitude of the antifibrotic effects that were reported by some studies is so big that actually who cares about finding patients that go from F3 plus down to F3 minus, right? The effect is huge. So I think categorical biomarkers such as the NASH CRN that we thought would not be adequate to evaluate these studies work extremely well when the drug is strong and works well. So I think this is a lot of very good news for patients. And I don't know what will be in the coming months the position of regulatory agencies when it comes to keep or not to keep biopsies as part of the recruitment. My take is that we are currently in the generation 2 of clinical study designs. Generation 1 were the very first studies, the Intercept, the GenFit, the Falcons at BMS, the Gilead. Pathology was there. We had one pathologist at best instead of a panel. So these were generation 1. We are currently witnessing the results of the generation 2 studies where pathology has significantly improved, where there is a cohort of biomarkers to enrich the understanding of therapeutic interventions in NASH. And I think we're going to see pretty soon a third generation of studies where non-invasive tests will play a major role.
Now, when you said major role, do you mean to the exclusion of biopsy?
Possibly. Yes, we don't know, but I think we're going to we possibly are going to have to see them happen, and it's a very good news.
I think for a patient. And clinical trials, On the way there, one of the things that you commented on that I think is important is that the NASH CRN 4-point scoring system is, in fact, its own problem. I forget who, but somebody published last week a study using the PATH AI ML system that went back and looked at the semaglutide data that had previously been defined as having no significant effect on fibrosis and said if you change it to a linear variable, the effect is about half a grade. And in fact, it's demonstrably significant over the same time. So the ability to do the kinds of things that you do or HistoIndex or PATH AI does will have some real value in generation 2. Now, tell me what that means for generation 3.
First of all, yes, in generation 2, having a continuous score that resolves what happens within categorical stages will be useful. Always. And in particular, if we are looking for a moderate antifibrotic effect, if you have a GLP-1 and you are trying to see if that intervention on the short term impacts fibrosis, you need biomarker with a high detection threshold. So our automated methods can work. I think the same questions will apply when we will be studying combinations, right? If compound A and compound B are analyzed independently and then together, what's the difference in the fibrotic effect? You are going to need techniques with very high detection thresholds. But with compounds that have such a big effect as the one that were presented at EASL, I think categorical stage will demonstrate that the intervention works.
Yeah, FGF21s, for example.
Correct. Yeah, absolutely. The Falcon 1 and Falcon 2 studies, we analyzed them. Our data didn't demonstrate that there was a superior statistically significant effect. So same conclusion as the categorical stages. But what we presented 2 years ago at AASLD is that we have been able to identify phenotypes of fibrosis that changed. And those phenotypes, when we put them together in a score, we see that the score demonstrates a statistically significant effect. In other words, and dose-dependent effect. In other words, there is a favorable remodeling of fibrosis, but not sufficient during the duration of that study to move patients from one stage to another one. So either patients don't respond well or strongly or quickly, or the drug is not potent enough, but it works.
So let's go on to generation 3. Your point, I think, was that for drugs with profound effect, FGF21, will there be drugs? You say combinations and moderate effects might still need biopsy. Some of the most robust effects probably will not.
I'm not sure. I think these are open questions there. I think the first question is, what will regulators go for? What we see, and you know that very well, we see retrospective studies with non-invasive biomarkers with a significant amount of data that show us that a non-invasive test could be considered as primary endpoint for the studies. We see that happening. Our community will have to explain to FDA, as the community has done in the past, to convince FDA that NASH serine stages were the right endpoint for interim approval. They will have to do the same for non-invasive tests. The case would probably have to be very strong for F2, F3 moderate fibrosis patients. If we're talking to severe cirrhotic studies, I think the case will be easy to put. So perhaps a stratification between severe decompensated cirrhosis studies and those F2, F3 studies. I don't know. What I see for sure is that as exploratory endpoints, and definitely in the context of phase 2 studies, to really understand what's going on and enrich what we learned from non-invasive tests, I think the use of biopsies will always be positive. And then there is another way of looking at this. If we believe that these treatments need a personalized approach, if we say this disease need to be addressed with a personalized medicine perspective, the fact that we have a biopsy is very valuable. In that case, we need to make sure that we get the most out of this tissue. And modalities such as oncology do that very, very well. So if we believe that a personalized approach is needed, then biopsies will play a big role.
Turning the question about 30 degrees, the question might not be whether personalized approaches are needed. It might be part that and part, are we capable of creating personalized approaches that bring value, right? In a world where you've only got 3 agents to work with, that's unlikely to happen. But as you get more agents, then in fact, you not only can do a better job of figuring out what phenotype or category a patient falls into, but then also what do you want to do about it, or how would you handle it differently?
And these hypotheses are not incorporated in any study that I know right now. But I think it's in the debate, should we or should we not keep biopsies in the context of MASH studies? The case could be made that biopsies will be useful not only to stage fibrosis and the related histological endpoints, but also to personalize or investigate possible personalized medicine approach or to stratify patients in different ways.
Okay, so let's play with that for a minute. If I think about the history of diabetes, there was a point at which everybody got metformin first line. And then after that, patients who did not do well on metformin or whatever point metformin did not work adequately went in different directions. If I think about that in the context of MASLD and MASH, maybe where I get to is a bunch of first-line therapies that I can give without personalization. But then if the first-line therapy fails, then at that point in time, I might want to conduct biopsies simply to understand enough about the patient on a personalized basis that I can prescribe exactly the right regimen for them. Is that kind of where I hear you going, or is that just my thought?
Well, I'm not sure I'm the best person because I'm not a physician. So it's always difficult to ask me how to manage your patients. Thinking logically, the first thing that I would say, okay, if you prescribe first-line therapy and you said if it's not working, right, how do you assess that? So it's going to be assessed based on non-invasive tests. Is that going to be assessed on ultrasound MRE? So I think that would be the first question. How do you assess and how long do you need to wait to assess that before you prescribe to something else. Because we're not talking about weight here. It's a little bit more difficult to measure. But I think if they are non-responders, or I should say slow responders rather than no responders, where there is urgency to address the metabolic etiologies, I think what you propose is very reasonable. But I don't see the avenues that could be proposed today.
We don't have options. I mean, back at oncology, how long did we have to be in oncology and how many different kinds of drugs did we have to have?
That's correct.
Or interventions in general before it made sense to personalize to that degree?
I think there are two considerations in terms of time. As you said, is how long did it took us to substratify breast cancers? It didn't happen overnight. And the other thing which a lot of people ask me says, how long will your biomarker, we're a digital pathology biomarker, how long will it take for this biomarker to become a surrogate biomarker so it can be used instead of the NASH CRN? Well, let's go back and take a look to A1C. It took a lot of time to turn A1C from a biomarker to a surrogate biomarker, and for all the studies to use that as a surrogate biomarker. So it's going to take a lot of time. We are currently running 2 outcome studies, retrospective outcome studies, to really be able to establish the link between our biomarker and clinical outcomes. One is an international study. The other one is a domestic study. We will actually retrospectively analyze the NASH CRN database too. Doing retrospective studies will accelerate the data that we generate. But having said that, being a surrogate biomarker is a extremely demanding value proposition that needs a lot of data. So we'll see if we can go as fast as this new generation 3 trials.
So all that's interesting, in fact, really interesting. And then the question becomes, so without giving away competitive secrets that you might not want to, how does it change what you do or how you think about going forward? Because really, the value in going to these meetings isn't merely to learn, but it's to figure out how to make better contributions to science and better contributions to commerce, really, at the same time.
There are maybe 3 answers. The first is we like to remain very, very collaborative with our clients, with the sponsors, because the question that we put on the table today, the sponsors put them on the table today, and our position, which I have reinforced in the team, is be very open, very true about what we can do, what we can't do. Because we don't want to mislead sponsors and their biomarker teams to say we have the solution for everything. No, we don't. We can definitely help. We have a roadmap to become a surrogate endpoint, but we are not there yet. So the first thing is we need to share and have a very trustable discussion with sponsors. This situation, it is what it is. And we have these discussions based on our data. And we are not feared. If biopsies go away, great. If my business needs to get reorganized, okay, I know how to do this. So we don't want to slow down the transition away from biopsies if biopsies don't bring significant value to these studies. Okay. So first of all, we'll find a way to get reorganized. And now let me tell you about ways where we can reorganize and where we are focusing. First, our biomarkers work as well in the context of preclinical studies. In the context of preclinical studies, there is a lot of activity trying to understand what are the impacts of the different classes of drugs on the phenotype of fibrosis. How do these drugs work? So that's number one. The second thing is, what are the hypotheses when you combine them? Are there ways to combine compounds so that the effect is magnified in a significant way? And the third arm is that we do more than fibrosis. We do all kinds of inflammatory and tissue injury phenotypes, which can apply to the study of antifibrotic compounds, but not only. So preclinical remains an area where we bring a lot of value. The fact that our biomarkers are also used in the clinic creates a translational avenue that is valuable. The other way to look is that fibrosis is not a liver-specific condition. There is fibrosis in other organs, and we are very active in Crohn's disease, where Crohn's disease is a fibrotic tissue because the strictures are basically very visible phenotypes of fibrosis. You could look at a stricture in a Crohn's Crohn's disease patient in the same way you look at highly fibrotic livers. And what happens is that the different inflammatory flares that happen during the life of a Crohn's patient create an underlying development of fibrosis. So there's fibrosis build up to the point it's visible and creates pain, discomfort, and all these symptoms. So there is a lot to translate from what we've learned in the context of liver to IBD and Crohn's disease. And this is an area that is fascinating, where we can bring a lot of value and where biopsies are part of the standard of care. You do them during endoscopy. So we don't have this problem of risky biopsy. So that's an avenue of a lot of interest. And there is a third avenue besides exotic fibrotic conditions where we can bring value, which is specifically in the area of oncology. You probably know that tumors are encapsulated by fibrosis. That's the good news. It's a way to protect the tumor to spread. But at the same time, that capsule is a barrier to all kind of anti-PD-L1 treatments. So you can't. And then the modulation of that capsule through combinations involving antifibrotic compounds, it's a way to really enhance the efficacy of therapies in oncology. So this is very new, but it's an area where we bring value. So to sum up, what we took away from EASL is, number one, we need to be very honest with our clients and sponsors and not be afraid of biopsies moving away. That would be a great thing. And that is not going to take us out of business. We have other plans that will maintain us in business to possibly help should we go through a generation of studies where biopsy tissue will be needed, not only for histological assessment, but also for complete phenotyping of patients.
That's interesting. Phenotyping really does take things to a different level. If you hang out with me long enough, you'll probably know, if you don't already, I say the same things over and over again. But you mentioned breast cancer. You know, in 69, when Nixon was president and declared that we were going to beat cancer, he thought there was one such thing as cancer and that he was going to find the magic bullet. First, we had to learn that tumors of different organs were different. Then we had to learn that breast, for example, had multiple kinds of tumors. And then you could kind of work back towards this quasi-integrated theory towards something like a PD-1 could treat in a whole bunch of different situations. It's intriguing to think that we can take the slides that exist in the biopsies that have been done in the past and use them not only to approve drugs, but also to phenotype patients. That we can start to understand, because I believe from the first time I walked into this disease, and late Stephen Harrison showed me a map of all the different ways that you could affect a liver in terms of fatty liver, that there were so many different paths to action that it couldn't be that this was just one disease.
Yes.
Right.
Absolutely.
So therefore, logically, we learned this with HCV also, but logically, phenotyping had to be a key to solution. And the idea that you can make more efficient use of tissue that we already have to phenotype it, if you know how to do that, is really, I think, interesting and important.
Yes, we do. And we've demonstrated, and we got manuscripts on that the histological phenotype of fibrosis really depends on its etiology. So if you have NASH fibrosis, or you have hepatitis C fibrosis, or PSC as well, or even fibrosis driven by alpha-1 antitrypsin deficiency, the phenotype of fibrosis is different. So you might have other etiologies of injury, of liver injury, that modify the histological phenotype of fibrosis. And now we're working on a side-by-side or an overlay of what we obtain from an H&E, which is basically a tissue panel, a single nuclei, single cell tissue panel, and overlay that with the fibrosis phenotype to look at the same time at the injury phenotype and at the fibrosis phenotype. In other words, we can make the difference between a hot septal bridge and a cold septal bridge. So I think through this approach, we will be also able to identify patients that can live with some level of fibrosis in the liver and patients that need to be addressed as well. So develop a prognostic of severity to be able to say, well, you're both in F2, but you know, this F2 here is the one that we need to address. There is some sense of urgency, whereas this other F2 probably needs to be addressed in a totally different way. So again, it's stressed the fact that fibrosis alone is not sufficient to manage the way you handle a patient.
So one more question. It strikes me that one of the reasons that alcohol and non-alcoholic steatosis were viewed differently is that there are differences in the injury and phenotyping patterns between the two.
Yes, absolutely. Yes, it did.
Has anyone taken a look at MASLD this way?
Oh, yes, we've done a study. It's a proof of concept study with collaborators in Japan where they had a cohort with purely metabolic MASH and purely alcoholic MASH. And we clearly demonstrated that the phenotype is different. And the test has an OROC of 0.95. So it's significantly different. The challenge is that now we have to validate this biomarker. And in order to validate, we need a large cohort where we know exactly if you have a patient that is drinking or not drinking. And I think the field is at this point to say, okay, what are the non-invasive tests that we need to use to really stratify patients?
Level of alcohol, yeah. Got it.
Yes. And I think the Madrigal study has this kind of biomarkers, and we are all looking at really understanding alcohol consumption.
So Matt, my friend, this has been fantastic. Thank you so much. Our conversation continues with Jeff McIntyre, Vice President of Liver Programs at the Global Liver Institute. So as part of this episode, as I said in the introduction, I'm here now with Jeff McIntyre, the Vice President of Liver Programs at GLI and a frequent contributor to the podcast. Just back from 5 weeks in South Carolina, so if you hear the accent, you'll know where it's coming from. Hey, Jeff, how are you today?
Bless your heart, Rogers. Good to see you.
Well played, my friend. Although you didn't say bless his point a little heart, which is roughly the equivalent of a profanity in certain parts of the Deep South. So welcome back to D.C. in the sweltering summer. How are you doing?
I'm good. It's been a super busy summer, and I'm very happy to be back and getting into the work and setting up what's going to be a very busy and productive fall.
Also exciting. So let's dive in. 2 simple questions, right? Question number 1 being, so what'd you take out of Milan? What were your takeaways? A, in general, and B, as they pertain to what you do.
I think, the main thing I take away from EASL this past May or June, I guess it was, is that there's more of an overriding sense of optimism. And I think, we've had these pop-up moments where we've had readouts about various medications that have come out. And there's always been this sort of low-level buzz around trendy things like, AI, there's new technologies, maybe there's new technologies being applied to diagnostics. The optimism that I see is really resting on the foundation of the survodutide, on the Boehringer-Ingelheim readout that Dr. Sanyal did. It really showed a strong response against fibrosis. And I think if the strength of its readout was quite amazing. But I think if you especially combine that with Madrigal's readout from last year and their approval process, you're beginning to see there's some optimism growing because there's been consistent good news. And I think folks are feeling optimistic about the Novo likely will have a readout at AASLD in San Diego this fall, this November. And so I think you're really beginning to feel more of a sense of, dare I say, grounded optimism. It isn't just kind of the, everything is awesome sort of Lego movie approach that it's all good, but you're actually beginning to see it backed up with these things that are substantive things that patients and the field and the industry can actually lean on to be able to take next steps.
Yeah, so, I get that. I get that totally. For reasons we may get into as we go through this conversation, I would probably add the efruxifermin late breaker to that and the pegozafermin data to suggest, which we'll get to why I say that specifically in a moment, but I think that adds to it, right? The platform of optimism. So it's interesting. One of the things we observed is when we did the preview episode for EASL, way back, feels like forever ago, but it was only May. One of the interesting conclusions that our entire panel came to, and this included Alexander Krag and Hannes Hagström and Jorn and Louise, and I don't remember if there's anybody else, was that a lot of the most exciting presentations that were actually not about the drugs, but about big data. And I don't think you can get to talking about big data if you don't have a solution for what big data teaches you. So you start to see tentacles that spring off the platform you're talking about. One is paying more attention to big data, right?
Yeah, and I think you see that sort of, the tentacles is a great metaphor for that. You see that begin to kind of reach out. That we've got, a couple of substantive medications that seem like they're doing good, big things. And then out of that, you're able to kind of collate this data from these things and what do you do with it? And as I said, for years now, there's been buzz about AI and machine learning. And I feel like a lot of that conversation has been ill-defined, and it's kind of also represented the larger conversation of which is, just all over the place about AI. But now is a place when we're beginning to have these big data sets where and companies are beginning to go, oh, okay, this is what that means. This is how we can do it. We can take these huge data sets and begin to not just analyze and not just be kind of like the philosophy student looking for meaning, but actually take something from it and use it for a deliverable and outcome that helps improve patients' lives in some ways.
Yeah. So it's interesting, not only the companies, but the health systems, because some of the talk that we had about big data was not just about what would it do in the context of medication. But if you take the data that exists in electronic medical records and you link that with repeat FIB4s, you can do a much better job of figuring out who's in trouble than you can with a single FIB4 or repeat FIB4s or far more expensive tests. That it becomes an inexpensive way to sort all that stuff out.
Yeah, I think so. And I think that also kind of expands to the other diagnostics. I'm sure that we could probably do a similar sort of thing with FibroScan as well to kind of see, who, what, where, when, and why on a larger set. And if we had the means to be able to do that nationally, internationally, regional sort of thing as well. And when we look at the big companies, your LabCorps and your Quest or your diagnostic companies, I think there's a real opportunity now to start tying that together with medication to hopefully deliver it. And at the very least, be able to tie it together to more sort of focused screening endeavors that we have as well.
Yeah, good point. So anything else that you took out of Milan, if the headline, if we think about this like a newspaper, right? So the headline is broader, stronger platform of multiple mode of action drugs. Then what's the second story?
I don't want to be too much of people know what a broken record is anymore, is a defragged MP3 file.
Yeah, there you go.
I don't be too much redundant.
Well, I just realized I said newspaper, and you know that we're both over a certain age because we both know what the heck I'm talking about when I say second lead. Go ahead.
So to not be too redundant, I do think there is this sense of optimism, and I don't want to downplay that. And it comes from multiple sources because we'll see that there are challenges here, there are challenges there. But even in the nature of our challenges, I feel like we have evolved into another level. It's like we've gone out. I don't know that we're out of the minors into the majors yet, but that we are definitely taking next steps for kind of greater challenges and stuff. We're engaging fibrosis in a way that I think, we've really struggled to kind of wrap our heads around, or for the field to wrap around how to best attack fatty liver disease. Also, to give credit, if you will, to the nomenclature change, moving away from non-alcoholic to metabolic, that it is holding the promise of showing now that we're having better and more conversations with the diabetes community, with, I think, frankly, the kidney community, with taking a look at the metabolic sort of aspect of this more so. And so I think there is that kind of undercurrent of the medication and the big data, but I also think that we're beginning to have more expanded and, dare I say, enlightened conversations in taking a look at what I would call the whole patient, but I think our clinician friends would call simply the metabolic state.
Hey, yeah, that makes a lot of sense. And not necessarily the way I had thought about it, so thank you. I take that as a gift. Second, what's interesting is that the other patient advocates that I've asked about the meeting have all talked about things having to do more directly with patient advocacy, whereas you've gone to patient benefit, if you will, but not in an advocate's way per se. Not here's what we do as advocates, here's where we're stronger. It's just here are the things that are happening that are going to help us.
Well, I'll be a bit of a homer here and say that I think it's just that GLI has a little bit of a different approach. I mean, yeah, we are patient advocates. It's the heart of what we do in this, but we're very focused on the outcome and the deliverable on this. And so we're very concerned about medication approval, for, if you will, which means we're very concerned about clinical trials. But to have that conversation without talking about patient access is artificial. And so for us, we're feeling, that we're having, as I said earlier, elevated conversations, that we're feeling empowered and able to have these conversations at higher levels with more diverse voices, with a larger range of stakeholders, actually move the needle here on this. I think that's a difference. There's kind of a talking about the thing, and then there's a talking about the doing of the thing. And that's not to have any criticism against my fellow patient advocates in such a way, but I think our perspective is really geared at getting people into screening, making sure that things are done from point A to point B in the drug approval process. We go into protocol review advisory boards already thinking about patient access down the road. And I think the FDA's recent statements and guidelines on diversity should reflect that. That shouldn't be just a clinical trial thing. That should be something that impacts patient access to medications down the road if it's done correctly. And so we're excited about that.
If the most important question for patients are, what can I do to help myself? Then that really has two issues in it, or what can the healthcare industry do to help me while I'm trying to help myself? And that really has two pull-throughs, right? One is what options are available, and second is what do I have to do to access them? So we've talked about the increased options. Do we have any insight on how this affects access or discussions about access?
I think the Well, I don't mean to go too meta on you, Roger, but I think the question is the answer there. It's kind of, the obstacle is the path, if you will, because we've seen with Rezdiffra, we needed a medication out of the gate. We needed something to get out there so we can now identify what are the issues we're going to have to go through. We can now run the traps actually instead of just theorizing about the traps. And this is helpful. I mean, we saw what happened with the Veterans Administration, with the VA, when they came out, and we've seen what we feared and has happened a little bit of the trickle down of the VA recommends biopsy for the diagnosis. That's a problem because suddenly we start getting a backlog of patients who have been prescribed the medication but don't have access to medication. And so that is the answer. Is the question you're asking is that, okay, now we get to ask different questions. Now we start to stress test this stuff and see where the mechanisms are that we need to influence on this.
Does the presence of more options have any impact on that?
Yeah, I think so. There's a little bit of a catch-22 in that, in as much that in the example of the VA, we don't want that. It has been, but we don't want that to be too precedent-setting. And so this isn't just us getting out there and fighting for Rezdiffra to not have biopsy, but by the VA doing this, it kind of creates this loophole, if you will, where private insurers now can require biopsy or recommend biopsy. And so when we get, if Novo comes through next time, if Survivitude comes next time, we were close to having this issue of biopsy kind of stamped out in terms of patient access. And so having more options gives us, it's important because it sets a precedent, but it also kind of gathers the troops, if you will, to have a bigger lever to push against this need for biopsy.
So if that's the first piece, then the second question goes, and therefore? So how does any of what you observed there change what you're doing going forward?
It allows us to work on the problems and the challenges that we have only been able to theorize about. So we can begin to get definitive answers. The Boehringer medication especially is something that allows us to implement the lessons learned from what we went through with Rezdiffra, and even more so with what happens with Novo in this way. I think that that's a significant thing. This allows us, the FDA's recent, as I referred to earlier, their recent guideline on incorporating more diversity into clinical trials. That's going to be a pain for companies. So they're going to have to be intentional in ways that they haven't necessarily been before. But if done correctly, as I said, with patient involvement, that can have a real payoff in terms of patient access down the road. It can create relationships. It can open doors. You can kind of have access to these communities now that previously hasn't happened in the past. And so to us, it defines the challenge much more better. We stop working out of a mode of anxiety and worry and consideration of all these possible things. And now we actually have tangible challenges that we can get answers for.
Okay, so there were two late breakers about drugs to treat MASH. One was fruxafermin, which I mentioned, and the other late breaker was actually about tirzepatide, which is on market right now, if not indicated, and also has some pretty good results. So I'm wondering why you're envisioning the issue more in terms of SEMA, whose results will probably not be as good as the tirzepatide results, and then survodutide as compared to tirzepatide. Is that about Lilly? Is that about it not having the indication? What's that about exactly?
Honestly, when you look at SEMA, I think it is the fact that we also have this conversation going on about anti-obesity medications in the U.S. right now as well, and about reimbursement for that. I think any conversation about that has to have that. It's not necessarily in the context of that, but it has to have that conversation attached to it. I think there are concerns that if we do get SEMA, and thank you for my pronunciation with just saying SEMA on that we could actually end up with kind of a cooling or chilling effect in terms of some of the medication approvals for liver disease specifically. And so we don't want to see that happen. We want to continue to push for specific medications for especially F1, F2 in this way. There's so much emphasis on the fibrotic state, which is part of why the Boehringer medication, I think, is really exciting in this way, is that if we continue to evolve in that way. Dr. Sanyal, in an interview I did with him for Global Fatty Liver Day, when I asked him about 10 years from now, what is a victory we'll be celebrating? What is the challenge we're going to have? And he says he thinks in 10 years' time, we'll be on the cusp of possibly getting rid of liver fibrosis as a disease. And that's glaringly optimistic, but I think it shows where the trend is in this. And if you combine that then with the antioxidant anti-obesity medications, that's kind of where my focus is coming from on that.
So if we're going to get rid of fibrosis, and first of all, I take the 10 years as being somewhat optimistic, given the original predictions about how long it would take to eradicate HCV. And here we are 30 years later, not having done it yet. So I appreciate both halves of that statement. But to me, eradicating fibrosis always had two pieces to it. It seems to me that number one is, can you put your foot on the hose, thereby stopping or slowing the rate at which patients flow into fibrosis, and then can get better drugs to treat them once they get there. You can see that both pieces are in place.
Yeah, absolutely. What's the quote from Archbishop Tutu, that at a certain point you have to stop pulling bodies out of the river and go upstream and see what's pushing them in. And so we have some of that going on here. And so that's where some of our optimism is coming in this, I think, is you're beginning to see that approach. And as I said, if you get the AOMs in there as well and get those reimbursed, then we're beginning to have, granted, it's come about in a fairly fragmented way, but you're beginning to have something come together. And that's where the shout out to now working around metabolic as opposed to non-alcoholic, I think, has really been promising and effective for the field in that way, as we're able to now gather these various different components and address it in that way.
Yeah, we agree. I've become increasingly convinced that while not every element of the rollout went exactly like I might have hoped, the name change in and of itself makes a lot of sense. And not just because of MedALD, but for exactly the reason you provide. And what makes that even more interesting to me personally, by the way, this is my interview, it's yours, but what the heck, is that the real benefit of SEMA as a simple GLP-1 is that will be easier to get used for diabetes and obesity. And if you talk about how to put your foot on the hose as compared to treat fibrosis, slow the rate of patients going into fibrosis, that's going to get done in diabetes and obesity. That's not going to get done with, I don't think you're going to see drugs indicated for F0 or even F1. I think what you're going to see is drugs indicated to treat the metabolic parameters, and that's going to be where the foot goes on the hose.
Absolutely. The goal here is to support and help patients, period. And we know, often said, it's cardiovascular events that are the big concern for just kind of simple fatty liver disease for F1 and F2 in that way. And so by having these AOMs out there, we're really beginning to help with that issue in that way. And so if we can help resolve that by any means necessary, which I agree with you, I don't think it's going to be a lot of F0, F1 sort of indications for this, or prescriptions, if you will. But if we get that resolved, then that allows us to focus a little bit more so on the more advanced states or where some of the other pain points may be in this.
Okay, so people complain that Americans use too many three-letter acronyms, so I'm going to put a definite. AOM is anti-obesity medication, and I'm going to suggest to you that the major issue may actually be AOIs, if you will, anti-obesity indications for medications that originally got approved for diabetes, right? I mean, I think that's probably more of what we're looking at, but not that it matters at the end of the day. Well, here's the sequel. The sequel is we're now going to start to look at some of the dual agonists going for their first approvals in obesity. We don't have a true metabolic AOM right now.
Yeah, I totally agree with that. But we can see that light at the end of the tunnel, potentially.
Yeah, I mean, I recommended 5 years ago to a couple of the dual agonist companies that they should hit obesity first, and at least one of them took me up on that. So we'll see what happens. But that would then become a true AOM. Not that it would necessarily get any more use than the multi-indication medicines where obesity is one of the indications of it, right? So, but I think all that makes sense. So what does that mean for what you do next year or over the next 3 years?
Yeah, that's a great question, Roger. I mean, in some ways it is, we continue the fight. You know, it is an elevated version of what we've always done, is that we continue to push with great passion and fortitude to get more patients screened. I think there are at-risk populations and marginalized populations that we haven't even begun to have that conversation with. And so we've got to get more people screened for liver disease. I think we've kind of held off in the past a little bit about trying to come up with the perfect non-invasive, if you will, and we're well past that need right now. We've got to get people out of the house that's on fire, whatever it takes. And if that's a multiple screening indicator, that's fine. I think we found that. Because now we have a medication that if they do screen at something more advanced, we can establish a clinical pathway for that to happen. I would be reft to say that we're working on these kind of two levels. One is that sort of serve the immediate need of the patient, get people, that individual, that family, that community screened, and begin to get them on the clinical pathway. For GLI, that's national and international in that way. For us, I think also we have, as I said, the potential of the readout in November for Novo. And so we want to take and examine what are the lessons learned from Resdifra, see what that happens with that, how we can best support that, how we can best kind of get that out there in the context of the anti-obesity medications. And there could be some kind of chicken and egg stuff that goes along with that, which, God help us, isn't too dependent on the American election necessarily, but could end up being influential there on that as well. And then I think the other thing that's important for us is that we really had a great event at the World Health Assembly with EASL, and working with EASL on trying to get liver disease and MASH on the agenda for the General Assembly next September is a huge priority for us. And it is not just so we have a shiny thing at the UN, but this allows us then to do more of the grassroots and grass tops works with what ultimately will be kind of the champion countries that we identify, that doesn't just help serve these underserved areas, but also then establishes a pipeline, if you will. It's almost like it establishes a foundation by which then we can have regulatory conversations in those countries to be able to move, better medication. And none of this happens without the screening on that as well. So it's going to be a busy year of both meta and micro.
Okay. I'm wondering where MIDI is. If we've got meta and micro, is there a MIDI somewhere in the middle? But forgetting about that for a sec, just two more thoughts as we get towards the end of the time available. Number one, the other conversation that's part of this episode is with Mathieu Petitjean of PharmaNest. And one of the things we talked a little bit was the ability to think about this a little bit like HCV and over time to move towards phenotyping. Not something we're going to do immediately. And that would allow for better dedicated use of drugs, probably better health economics, and easier to get things approved or paid for. I'd love for you to comment on that.
I will give the caveat that I don't know a ton about that. Immediately go off from that. But I think that is absolutely a valid goal. I think it's something that we have to be working towards in this. I think I've been more focused on the immediate than I have on the phenotyping of this.
Yeah, you kind of have to learn to walk before you can run the 100 meters. I get your point.
Yeah, and it's been more dictated by the circumstances here as well. But seeing that as a logical and even perhaps logistical next step, maybe even not next step, but the next step in this totally makes sense to me.
So here's another next step that actually I wasn't thinking about before I've been listening to you, so it just kind of popped into my mind. If you start identifying countries to deal with regulatory, do we think that'll lead to a fairly common result everywhere you do it, or will the results be different enough that you can then go to the second level countries and say, well, you got option A, which is what they're doing here, or option B, what they're doing there? Do you think all this evolves as a single global model, or do you think it starts to splinter a little bit?
Oh, no, it definitely splinters. No question about that. It's kind of like the Monty Python quote where the crowd chants, we're all individuals. It's one of those things where there is going to be a common thread, but we have seen, for instance, if globally people look to the FDA generally acts first. After that, it goes to the EMA. Generally, Europe is close behind that. After that, it begins to disperse out. And what we've seen, if you will, and I don't think I'm talking out of turn when I say this, if you see the regulatory models for, say, Latin America, if you look at Mexico and Brazil, then if you can get through those regulatory systems, and they tend to follow a good bit of the lead, although they're very much their own independent entity. They tend to, take into consideration, I'll say, what's happened with the FDA and EMA. And if it's good stuff, then that sets us up better in those positions. But then you tend to see a little bit more of a domino effect through the rest of Latin America. And it's not that there can't be issues that pop up in Uruguay or Argentina or, Paraguay or something, but it makes it easier for us to make the case in that instance. I'm kind of talking out of both sides of my mouth for your answer there, Roger. We are all individuals at the end of the day. So yeah, I think it does transfer in that way, but it doesn't mean that we don't take the individual country any less seriously in that instance. And then what we take from there, we're able to talk, with Philippines. China's kind of a thing on and of itself, which has to be engaged kind of in a separate way. And we begin to run into different issues there. One of the things that we've seen is for the acclaim that you and I have given over the benefit of the nomenclature change, roughly a third of the world's population isn't using that nomenclature right now. They're using MASLD and have a bit of a chip on their shoulder, frankly, about it as well. And so we've got to be able to engage those countries on their terms and in a culturally competent way that engages that disease, in the way that they talk about it. But the heart of it is still going to be metabolic, and I think that that's where the biggest promise of this is.
So then one more small question, right? You called what used to be known as International NASH Day Global Fatty Liver Day this year, which I think was, as much as anything else, as you explained it to me, in recognition of this MASLD thing, that if people are calling it by different names, then you can't just pick, as a global patient advocate organization, just pick one nomenclature, even if you'd like to pick sides. Forget whether you would or not. Over time, does the word metabolic work its way into the title of the day, or does it stay focused on on fat in the liver? Would you guess?
My guess would be probably not. I wouldn't say it as an impossibility, but I mean, it's a theoretical more so than anything else. When we talk about patients, or talk to patients, I mean, as you referenced, I was just in South Carolina. I spent the past 5 weeks up in a small Appalachian town there. And if you say metabolic to folks, they're not going to quite understand, or at the very least, they're not going to make the connection between metabolic and liver, if you will. I mean, people don't make the connection between diabetes and liver necessarily. And so if that were to happen, we'd really have our work cut out for us in this. And we find that fatty liver disease is something that is descriptive, it's simple, it's easy to understand. I know there are those that disagree with us, but our patients reflect to us that it's not stigmatizing. And in fact, we've seen that reflected also in Asian countries where it's the issue is that it's the opposite of stigmatizing. It's actually an ideal that they're trying to live up to. And so that's why we've stuck with fatty liver disease is it opens the conversation, and it opens the conversation in a way that is kind of where and how patients talk about it.
And then to go back to a phrase you've used in this conversation, metabolic then becomes the meta discussion.
Yeah, very much so. I mean, and I feel like we're trying to find a solve for liver disease, and it's for MASH, MASLD, MAFLD, whatever it is. It's that fat on the liver that goes fibrotic and then cirrhotic, and it's that process. And once we talk about metabolic kind of without the other context, I think a lot of folks then begin to get a little confused by that it can be confusing. But if we talk about fatty liver, that's a very concrete, operationalized sort of thing that people can actually visually imagine much more easily. It leads to an easier conversation where we can go, this is that, thus get screened, get treated,
Okay, so we're 2 minutes past the end of the time you told me you had available, and that answer led to a question. So if I can ask you one more, and I promise this will be the last one. Does that extend to Met-ALD as well?
I don't know, honestly. I think the Met part of Met-ALD will probably not be recognized as much as ALD still is for patients. I think that's a fairly clinical designation right now. I think people are still going to see, I mean, we still suffer from the stigma of alcohol disease on the liver, and liver disease is being related to alcohol, period. And so the notion of it there being a metabolic sort of thing, just for patients, I don't think that's going to stick a whole lot. I think it's an important clinical designation. I think it's going to be very important to the science and to the researchers in that way. But for patients, that phrase alcohol liver disease is going to be so prominent as to the fact that there's a metabolic aspect to it. It'll help them when they're in the doctor's office maybe a little bit, but it's not going to be a determining factor.
Okay, thanks. What else should I be asking you about that I haven't? That's the first question I've asked where your answer hasn't gotten me to another question I hadn't thought of. What else? Is there anything I should be asking about that I haven't yet?
There's so much, Roger. I feel like I'm a little brain numb in terms of all the things that are going on. I'm still catching up on emails and stuff. That's all that comes at the top of my mind on this right now.
So then let's agree to have another conversation on this so we can record in 3 or 4 weeks.
Awesome.
We'll see where you've gotten to from there. Jeff, thanks. This has been fantastic.
Always, Roger.
And our third section is the question of the week, in which Mike Betel and José Willemse respond to 3 questions, one each from patient advocate Bruce Dimmig, transplant survivor Sue Green, and public health executive in Argentina, Constanza Enrico. After they're done, I'll be back with this week's question of the week and a discussion of how we intend to use it further, and then our business report. So hang in there. So with that, let me just read answers. The first answer came from Bruce Dimmig, identified as a patient advocate in Arizona, U.S., and a patient as well, who writes, as a patient, I believe that hepatologists need to be honest with the patients, and the biggest thing is to actually listen to the patient. We know our bodies and don't want the doctors to dismiss the symptoms, etc., that we're having. I know that it's hard to find the time, but it would be great if there was a way that appointments could be longer. Another item is to provide a more team approach and have the hepatologist consult with the patient's other specialists, such as cardiologists, nephrologists, hematologists, etc. Okay, that was Bruce. Second respondent, Sue Green, who identifies herself as a transplant patient, replied, I believe that there are 2 areas that need to be addressed in terms of communication between patients and providers regarding MASH. The first is pre-transplant, the second is post-transplant. Many doctors are not educated or knowledgeable about MASH, how to identify it, and how to help patients manage their illness. There are a large number of people who've been diagnosed with MASH who do not understand how serious their illness is and what they can do to manage it. I once asked my online support group of people dealing with what was then called MASLD if their doctor had educated them about what to do now that they'd been diagnosed, and 75% said they had no clue what to do. Their doctors had not mentioned any diet or lifestyle changes. Doctors tend to know very little about helping patients achieve lifestyle changes. If they cannot prescribe a drug, they can be at a loss for what to do, choose to do nothing. She goes on this subject and then goes on to say many patients would benefit from a referral to a health coach. Instead, the whole issue appears to be dropped. Her second issue has to do with grave concerns about a post-transplant issue. It has to do with the fact that there have been no new immunosuppressants developed since 2011. There are financial and FDA-related issues preventing innovation there in this area. However, many post-transplant patients struggle with immunosuppression, the side effects, and the fears of possible disease from the immunosuppressant. Some patients feel their quality of life is disturbingly bad. Many feel they were told they'd had to be on immunosuppressants for the rest of their lives, but were not given a very good idea about how difficult the struggle could be. I know that people in the field recognize immunosuppression is a difficult thing to deal with. However, I feel it's minimized prior to transplant, not responded to after transplant. She goes on from there, but I think these are the major points. And she would like for patients to be better educated. She says it won't change anyone's mind, but it will make them better equipped to cope. And finally, once people go on immunosuppressants, she's concerned that providers don't really want to deal with the issue. So patients don't really know how to handle. The third answer came from Constanza Erico, who works in the public health ministry in San Juan, Argentina. Her response came in Spanish, so this is a Google English translation that I think is reasonably good based on my limited Spanish. She wrote, I believe that working in multidisciplinary workshops, or if you will, centers, where we empower patients about healthy habits, adherence to indicated medication, and physical activity are all of the utmost importance. These three pillars must be understood by the patient and objectively expressed by health professionals. All members of the cardiometabolic team must speak the same language, and instruments such as pre-printed and digital clinics. I think what she's saying here is that having pre-printed materials, digital reports, and things that the entire team uses in the same way will help present information to patients in a reliable way. So 3 different perspectives, right? One fundamentally about teams, one about information to patients and what providers do and don't know, and one about teams, but really kind of in a different way. To start, let me ask either of my 2 panelists, take a minute or 2 and tell them what you think. Pick the topic of your choice.
I'll jump in. I think there's a lot to unpack here, and I love the fact that people took the time to respond and share their feelings and their thoughts about this important issue. What I come back to is something we've talked about before, Roger, and that's the importance of tailored care. That means that the physicians really should look at each patient as an individual, because as really all 3 of them, the answers said, the responders said, that patients are individuals and need individual care. And maybe if the physicians had more time, which Bruce talked about, and it's probably not a possibility, they would be able to be more curious about that particular patient and be able to ask the questions that could bring out the needs of that particular patient. So I think in that respect, they're all tied together. And, you know, with Sue Green's point about the lifestyle piece and education, there is, I'm not want to put the onus on Madrigal as an example, but being the only drug approved right now in the US, maybe their team, when they're out with physicians, they could also take just a little bit of time. In pharma, we used to call it like a second detail, but after the talk about the drug and the care, just to mention the lifestyle side of it, so they can find out if their physicians or physicians who refer to them have everything they need with regard to the education needed to support patients in lifestyle management. I think that's really important as well. So I think that covers it. So my takeaway is be curious, care more, and offer the support that's individualized for the patient.
I will drop in here. I think that all 3 questions, well, we could fill an hour with each question. And what triggers me is the question of Bruce Dimmick, that he said, well, I wish there was more time, but it's not always a matter of time. When I say to doctors, please ask a patient, well, how does it affect your life? Say, yeah, I want to, but time, there's no time. So it's just another way of discussing things with your patients, and skip all the less important things. Ask your patient, are there some issues you want to discuss? I have some, but do you have? And then you can move on with issues that are for patients very important, and listen more to patients. But the excuse of there is no time, I'm fighting against that, because even if you have less time, it's a matter of an attitude, how to see your patients and ask the right questions and open the floor for your patients. So it's not only a matter of time, but also a matter of attitude. And I think that's something that we can change very easily. Well, it's not easily at all, but doesn't cost money, and only just another attitude.
Hey, José, you said before, and you should reiterate what you said, even requote yourself, that listening is different from hearing.
Oh, yes. If a doctor says, I hear you, and then I said to the doctor, but you don't listen. So, and that is really a difference. And that's the attitude I was talking about. Yeah. definitely.
Let's start by positing a couple of things, right? There isn't more time. However, there are protocols and priorities. And when I hear I don't have the time, what I hear is the list of things I have to do according to guidelines or organizational protocols is going to take all 15 minutes I have. So at that point, your comment about ask a patient-centered question first is something they may feel they don't have time to do. There's an old organizational management saying that people do what's inspected, not what's expected. So if what's inspected is that I'm doing everything I've got to do on my checklist, and that takes all 15 minutes, then no, I don't have the time to think about the patient. It would be helpful maybe under a program like motivational interviewing, or maybe in a different way, to work with organizations to make sure that part of that checklist is José's question. That in fact, if you ask this question and you get answers, then that's worth any 4 other items on this list. For example, I don't know how you do the algorithm for that, but if you can't get it into a protocol, it becomes hard for people to do it, to motivate people to do it, unless they are motivated that way. And then it becomes hard to train on. Mm-hmm. If you can get it into the protocol, then you can start to train.
What about, like José brought up before the fact, her experience is that physicians often, because they're either reviewing the file or keeping track, as you just said, of what they have to record for the medical record, that they're actually not engaged that much of those, let's say it's 15 minutes, with the patient. They're looking at the screen and sort of they're checking off their boxes, but they're not actually speaking with the patient. Is that how you feel?
Yeah. Exactly. And it is possible. I know doctors, and Tom Yo, he told me last time when we were online that he's really doing that. The doctor said, well, let's first do my thing. That's all the test results. And well, these are my highlights, and this is what I want to talk about. And then he switched off his screen and said, well, let's talk about you. How do you feel today? And Tom is doing that now. And he was so surprised about the interaction with the patients is so much better now, and that he got so much more information, because very often you have always information behind the information of your patient, and that can be done in the same time. And it is about the patient and not only about the liver, and that's exactly how people very often feel. It's about my liver because it's only about test results and not about me, but it's me who had the liver problem.
Yeah, we are more than our livers in many different ways. I'll tell you, I think it's a good rule everywhere in life that when people want to have personal conversations, they should turn off their computer screens and look folks in the eye. Or if you're on the spectrum or seriously ADD, I'm not on the spectrum, I am seriously ADD. Not always easy for me to look people in the eye, but if I have a computer screen on, my eyes will go there. And that will diminish the sincerity of the conversation. That might be another one, right? Mike, why don't you share what you just did and why?
I shut my screen so that I could pay attention, realizing that, of course, I can't participate.
Well, yeah, although the nice thing about this, right, and people don't know this if they've never been part of the podcast, although we only present in audio, we record in video. And the reason is I can look José or Mike right in the eye as I'm talking to them and be on the screen at the same time, which might work actually for telemedicine, but doesn't work for in-office visits.
You say you have a face for radio, right?
I do have a face made for radio. It's true. Particularly my nose. I very much have a nose made for radio. Yeah. So other than those couple of thoughts, right? You'll say the need to get the doctor to ask patient-centric questions, at least one patient-centric question, and for the patient to feel that the doctor is centered on the patient when asking that question. So if I'm looking at my computer screen, I'm just staring in a different direction. It's not going to work. If I'm looking at my computer screen, which is 45 degrees off the patient's face, and I'm typing on my keyboard, the patient's going to know the question isn't entirely about them. But if you turn off the screen, ask an open-ended question, then that's a tone.
No, and people of patients have really the feeling that the doctor at that moment is there for them, and that's so important, and that you can talk about more issues than only about your liver tests, because it's having a liver disease. I think any disease, especially chronic diseases, has a big impact on your life, and it's nice to hear how your liver is doing, but it is about the impact on your life you want to discuss, and your fears and what you wonder, or your happiness because it's going well. That's what you want to share as a patient, and not the level of your ALT or AST. Very important for the doctors, and I always tell doctors, keep on doing that, but that's one part of your consultation with the patient.
So, Mike, if you don't have a comment, let me throw out one more thing that came out of those three answers. This is from Sue. You'll say what you're saying, I think, becomes particularly important to answering her question about immunosuppressants. If immunosuppressants lead to patients being depressed or their quality of life being decreased and they have to be on it for life, then there are people who are motivated and will find a way to overcome that. We have some fantastic liver advocates who are transplant patients, and they live active and engaged lives, despite whatever else. But then there are people who are dragged down, and the question becomes, what kind of support should the healthcare system or the physician be providing to those patients, A, so we can figure out who's really being dragged down badly, and B, how to help them rise up again? Thoughts about that?
Yeah, well, I think about, and that's not only about immunosuppressive medication, but also for other medications, that people are not aware of the side effects at all. And it helps if you warn people like, hey, listen, this can happen to you. Depressions or feeling depressed and just the brain fog, all issues that people have and think that they are, started to have mental issues as well, besides their liver disease or besides the medication they have to take. If you are not aware that this belongs to just the treatment or your disease, you have an extra worry. And I have these conversations very often that people say, I don't feel well and these are my problems and I have to go to another doctor because maybe, dementia started maybe. And then I always say, I'm not a doctor, and especially not by the phone. But if the doctor can't find any signs of dementia, it belongs to your disease, or it's one of the side effects of your medication. And how often people say, you're kidding me. And I said, no, that can be true. And I always wonder, why is there no more information about the impact of side effects? And if you know that this is a side effect, it's better to deal with. And it's not makes your life happier, but if you can deal with something, then it's much easier in life. So it's also a matter of information.
Mike, what are you thinking?
It's really a best practice to inform patients of adverse events that might be experienced when they're on a treatment. So I would say that's something that patients should expect with regard to the education that they receive before they start a treatment.
Yeah, I agree with that. I do have one other thought, which is that doctors aren't trained empaths. And if we're talking about psychological effect of medication that you have to be on for the rest of your life, the ability of the doctor to steer patients to support groups with other patients so they can share experiences and develop, if you will, patient best practices informally, and also have someone who really gets what you're talking about. It feels to me that that's a place where it's hugely important, one of several.
Yeah, the point is that not everywhere are support groups, but you can set up something in the hospital. A lot of transplant centers have, but sometimes people have a little bit fear to go there or to ask their questions because they think it's, maybe a little bit dumb, but it isn't at all. Or they don't realize what their symptoms, how they are related to the medications. So they don't think about, hey, I need peer support. That is very helpful. And I want to bring up another issue, and that is adherence. When you're such a long time on medications, adherence is an issue as well. Nobody talks about that you have to be aware of that, and that if you want to quit your medication because, I'm all right, and things like that, that's also one of the issues there's no information about. People are not warned about that. And if doctors are not so qualified to give that information, we have the nurses in transplant centers. They can do a better job on that issue.
I think that all makes sense. The one other thing I'm wondering about, though, is the idea of things like what Sober Livers is doing, Project Hope, right? Which is, we're in an era where I think we can put a lot of this stuff online, on patient-to-patient basis. Folks might be concerned about going to their own hospital's transplant center, or they might be concerned about saying anything negative about their experience in a group like that because they don't know who's listening. So, Mike, I'm going to ask you this one. What are the impediments from someone like Fatty Liver Alliance setting up a Canada-wide group, besides the fact that you probably have to learn to speak French, but in all seriousness, set up a Canada-wide group, one in English, maybe one in French, that becomes patient supporting patients through all this? What impediments would you see? Does it make sense to you? And why or why not? And what do you see as the impediments and benefits?
Yeah, in Canada, there isn't one that I'm aware of. I know there's a few in the States, like National Knowledge, as an example, has a group that they speak with and they follow. I tried a couple of times to do something through Facebook, and there was like a few people who started, but then it didn't really follow through. I think to make it effective, you have to have somebody dedicated to it on an ongoing basis so that they can keep seeking new engagement. And that's a whole whole project on itself. So I would say, I think it's really important, especially working in conjunction with the treaters across whatever country you're in, so that you get referrals back and forth and you can work with the patients. But it just needs a dedicated resource. And that was a challenge for me from Fatty Liver Alliance's perspective.
Yeah, which means it needs a funding source, huh? Where I come from, dedicated resource equals funding source.
If there was one person, that's their whole role, was to recruit, engage, and educate in a discussion two-way format patients, it would be fabulous. But you're right, it's completely resource related, because I'm sure there are people who would want to do that kind of role.
Mm-hmm. You have any thoughts on that?
Thanks for bringing that up.
No, and I agree with Michael. It's very difficult to find people who continue the whole process. That's one of the main issues also for patient groups.
I totally understand that one. Okay, so I want to thank the two of you for being with me today. I want to thank Sue Green, Bruce Dimmig, and Costanza Enrico for being good enough to send us these questions. And to our listeners, we're finding more and more ways to get the question of the week out there. We'd love for you to answer, because if you do, you will hear this conversation, you will be mentioned, and you'll be invited to participate. So with that, let me say goodbye to my friends, and I'll see you guys soon, and I'll be back with a business report in a couple of minutes. Thanks. Bye-bye now.
This week's question of the week is sponsored by Madrigal Pharmaceuticals.
And here's this week's question of the week. During the episode, Matt and Jeff discussed what they considered key lessons from what everyone has described as a pivotal meeting, EASL Congress 2024, and how those lessons have changed their view of their strategy and or their work. How about you? How have presentations or discussions from that Congress and the key lessons you learned reshape your thoughts about your work?
And now for the Season 5, Episode 24 Business Report.
Slava Ukraini, glory to Ukraine, Heroyam Slava, glory to heroes, and also Am Yisrael Chai, the people of Israel live. Last time we did one of these, I said, and I'm quoting myself, we can only hope that the tone and substance of the Republican convention does not give encouragement to the enemies of democracy, end of quote. So much for that. Whatever his other strengths or shortcomings, and I'm not going to endorse candidates or pick sides, J.D. Vance is, as opposed to supporting democracies outside the U.S., and particularly Ukraine, as anyone in the U.S. Senate. In the so much for that category, Bibi Netanyahu came to Washington this week and spoke in a tone that was hardly conciliatory, discouraging on both fronts. But life goes on. With that said, let's remember and respect the brave Ukrainian fighters and residents, remember and wish for the safe and speedy return of the many remaining Israeli hostages, and remember and pray for the safety for the Gazan residents whose needs are poorly served by their government's approach to this situation. Heroyam Slava, glory to heroes, and also Am Yisrael Chai, people of Israel live.
Next week, we move beyond EASL to discuss a major recent article.
In episode 11 this year, Michael Charlton alluded to a group of U.S. key opinion leaders working out recommendations on treating with resmetirom. That paper, titled An Expert Panel Recommendations, Practical Clinical Applications for Initiating and Monitoring Resmetirom in Patients with MASH-NASH and Moderate to Non-Cirrhotic Advanced Fibrosis, pressed in Clinical Gastroenterology and Hepatology on July 20th. Maru Ranella, who is corresponding author, and we hope one or two of the other co-authors, will join us next week to discuss this very important paper. It's a big deal. I'm really excited we're having this discussion. We're still interested in having a major NIT episode, which was the original plan, but that will come sometime later this summer.
Question of the week. Imagine the possibilities.
As you heard in this episode, we not only want episodes to our questions of the week, we want to explore them and invite questioners and panelists to join us in doing so. From time to time, people ask me, how do I get on your podcast? Easiest way? Provide a thoughtful answer to a question of the week, and ideally get one friend to do the same. 2 serious answers? It's in the program. Serious.
Office hours continue next Wednesday, July 31st, at 11 o'clock Eastern U.S. time.
After a week off for my birthday this past Wednesday, don't ask my age, office hours are back next Wednesday, July 31st at 11 a.m. Eastern, which is 5 p.m. in Central Europe, 4 p.m. in British time zone, and 10 o'clock a.m. in Chicago. I will publish the Google Meet link for this in next Monday's newsletter. There'll be in every newsletter thereafter. And I will include a question I'd love to discuss with any and all interested parties.
No vault this week.
We've been a bit behind schedule getting this episode out the door, so we're going to skip the vault until next week. We've got plenty of content in the interviews and in the question of the week. That's it for today. I want to thank Matt, Jeff, José, and Mike for stepping in to pull this week together. Please take a listen to our question of the week, and now that you know where it can get you, reply. If you're a LinkedIn follower, check your email or messages for an invitation to subscribe to our weekly piece and for how to access my weekly office hours Wednesday at 11 a.m. Eastern U.S. time. As I discussed already, next week will be really exciting for us. Until then, stay safe, surf on, see you on the podcast. Bye-bye now.
This week's question of the week has been sponsored by Madrigal Pharmaceuticals.
Have any questions for the surfers? You can send them to questions@surfingmash.com and we will answer on the podcast or website. Your question might even wind up being the question of the week.